you for coming tonight. So let me introduce our speaker for today. So we have Professor Ewa Paluch, who first graduated in physics, then did a PhD in biophysics at the Curie Institute in Paris. In 2018, she was elected chair of anatomy at the University of Cambridge, and she's the 19th professor of anatomy here, and is the first woman to hold this chair in this 300-year history. Prof Paluch has received a number of awards, including the Fields Medal from the British Society for Cell Biology in 2017, and the Blavatnik Award for Young Scientists in 2019. So the Paluch Lab combines approaches from biology, physics, and engineering to investigate cellular shape control in healthy and disease conditions. Today she will discuss how multi-disciplinary approaches are becoming increasingly central in the life sciences and highlight some of the group's findings on shape and form in biology. So let us welcome Prof Paluch.
Thank you very much, Kathy, and thank you for the invitation. Should I use the microphone?
No, we can use you.
Okay, if it goes down, please wave and tell me use the microphone. Thank you. Thank you for the introduction very much. Just want to point out that 300 years ago there was no way that there would be a female chair of anatomy, so it's not as big an achievement as it sounds to be the first female one. So Jessie asked me when I asked her what kind of talk I should give, so she told me that maybe something a little bit about science and a little bit about how and why we combine physics and biology, and about how and why I made different decisions in my career would be good. So this is not going to be a very data-heavy talk, but more like a talk about my career path in a way, and the questions we've been trying to address, and how the lab is really trying to not do physics and biology side by side, but really combine approaches from physics, biology, engineering, computing, and put them all together to understand this fundamental question we're interested in, which is how cells, and I will be talking exclusively about animal cells. That doesn't mean that plants don't matter, but they have very different ways of controlling their shape. How cells control shape. And I don't want to exclude bacteria either, but they also have very different ways of controlling their shape. But first, before I go into that, I just would like to tell you a few words about how I got into physical biology in the first place. And actually, it was not a very direct path, because in fact, I came to it from a very sort of extreme outside. I actually initially in undergrad studied mathematics because I was really attracted to like the abstraction and beauty of it, which I suppose some of you can relate to, and also because in France it's kind of the standard way of trying to avoid making any decision, because in the first years of studying mathematics you actually do mathematics, physics and chemistry, so you don't have to commit to anything too strongly. And so after 2 years of doing essentially a major in mathematics, I realized that actually, well, it was very beautiful and abstract, but maybe a little bit too abstract. And so I switched into physics, still trying to understand the basic principles, or at least with the ambition of trying to understand. I'm not actually claiming having contributed to it very much, but with the ambition of trying to understand the basic principles of how the universe works. And as I think many young physics students have that ambition probably. And so my first internship I did at the Observatory of Paris-Meudon, studying stars made of quark gluon plasma, or doing some very minor computing in a very big study of how fast a star made of quark gluon plasma would turn. And after that, the year after, that was in 1999, I was actually a summer student at CERN in Geneva, where I still worked on quark gluon plasma, but more on the particle side of things, and again doing some rather minor coding on trying to understand what happens in the accelerator when quark gluon plasma is produced. So that was very exciting. I must say that I barely remember anything about it. I couldn't explain very much about what I did in these internships at this point. It's been a while. But somewhere during that time, I realized that actually this was maybe still not quite exactly what I wanted to do. And the revelation moment came when I was writing my internship report in the library at CERN. And I was looking at the bookshelf where the PhD theses of the different people were who had just finished their PhDs there. And I came across this one PhD, which was about 500 pages. We had two actually volumes. And the title was Calculation of the 17th Decimal of something, some very important constant. And that was a moment where I thought, I do realize why this is really important. I'm really glad that there's people studying this. And I actually felt very excited when the Higgs boson was discovered, but this was not for me. And the experimental part of it has had similar things which didn't appeal to me very much, It is very exciting and crazy from an engineering perspective to work in such an accelerator, but you're part of a team of 200 people, and I realized, by having tried to do this, that actually I like much better working in teams of about 10 people. And so this kind of fundamental physics was maybe not, but also not one person, so not theoretical physics. And so this kind of fundamental physics was maybe not what I really wanted to do. So I was a bit lost, and that's where, and I think that may be a message for many students here, is that quite oftentimes the decision you make in an academic career come down to who you meet and who you talk to. And so somehow I had a friend from my physics course who was 1 year above me, who had just joined a master's degree in Paris, which was called Interface Physics Biology. And he was telling me about what they were doing in that master's, and I thought that sounded just amazing, because what they were doing is essentially providing training in biology to physicists like me, who wanted to try to apply some of what they had learned in physics to questions in biology. And so after talking to him and realizing what this master's was about, I decided to apply, and from then on I basically became interested in trying to understand still the general principles, but of something a little bit somewhere in between in terms of scales, in trying to understand how actually cells work, which is obviously a huge question, but more specifically, how cells work from a kind of biophysical perspective, which is still a huge question. And in terms of what specifically I ended up doing in terms of research, and I'm still doing now, again, it was a matter of encounters. And I was very privileged in my master's to come across these two people who still work, actually Michel retired now, at the Curie Institute in Paris. So the Curie Institute is a little bit like Cambridge in terms of being daunting, just a little bit smaller. So you have like Pierre and Marie Curie sitting here looking and checking if you're working enough or if you're having your coffee break. And obviously it's an amazing place in terms of history and what happened there. But also it was, back then, one of the first places in Europe where physical biology was being actively applied to cell and tissue. So not at the molecular level, but rather at higher scales. And I was very lucky to be co-supervisor in my PhD by Cecile Sykes, who is a physicist, comes really from condensed matter physics, but is really working now at the interface of physics and biology, but more on the in vitro side, looking at cytoskeletal networks in vitro. And Michel Bornas, who is a pure biologist, but he's always been interested in discussing with physicists and has had over the years many collaborations with people coming from physics and mathematics. And they are both extremely inspiring people. I owe them a lot. And Cecile is maybe most inspiring by her like can-do attitude. She's just not afraid of anything. And Michel is like a walking encyclopedia. I think he's read all the papers that have been written in biology since 1910, probably, and or maybe even earlier. And he's a fascinating person to talk to because he really, really knows a lot and thinks about things very deeply. And one of the things he taught me is that it's always very useful to read the old papers, and I've been trying to apply that within limits because there's only so much time we have, but it's sometimes very interesting to go on a tangent and try to find all the papers that have been published in the very early days of the topic you're interested in. Quite often it's quite inspiring. And so working with them, what I became interested in, and I'm still interested in, is the question of cellular shape control. And this question is quite fascinating and important, maybe for obvious reasons, because, well, as you're all aware, we all come from one rather boringly shaped cell, which is the fertilized egg, which in most species is pretty round. Sometimes it's a bit oval, but nothing too exciting. And then the cell divides many times, and often, not in all organisms, but in many organisms, the first division still conserves this kind of round shape. And then at some point something happens, cells start differentiating, and as they do so, to become the specialized cells in your body, which will do what they have to do, as they do so, they usually acquire a variety of shapes. And then in the adult organisms, they look actually very different if you look at cells in different tissues. Neurons are quite extreme. They have these very, very long axons, which can be meters long. Cells in epithelia look like little tubes. Muscles have also very specific shapes. Red blood cells look still different. And then migrating cells like neutrophils and the white blood cells have very floppy shapes. They are very deformable, and that's actually quite important for what they do because they need to go scan your body and in search for infections and pathogens. And also, and that's a rather bad example of shape changes, but one key feature of metastatic cancer cells is that they are also really quite deformable, and they change shape quite dramatically when the more metastatic they become. So understanding this question is actually really of fundamental importance, both for development, for the physiology of the adult organism, but also for many, many diseases. Now, what's exciting about it from a physicist perspective is that actually shape fundamentally is a matter of mechanical forces. Doesn't matter if the shape of this table or of me or of a cell inside my body. In the end, what makes a shape is what forces are exerted on the surface of that object. And that's something that actually Darcy Thompson, who was, speaking of old papers, who was a very prominent and scientist interested in the diversity of different things. He was a biologist, but he was also one of the first, he was involved in one of the first efforts to preserve species in danger, for example, back then in the early 20th century. And in 1917, he published this book, which is called On Growth and Form, which I don't know if it was very much read 50 years ago, but in the last 20 years it became a sort of Bible for morphology or morphogenesis interested people and developmental biologists in general, because it discusses essentially how shape and patterns can be acquired in an organism. So obviously it's not very molecular because it's from 1917, but it raises a lot of important questions and proposes a number of interesting, some of them wrong, but that's okay, approaches to these questions. But also, he was one of the first people to actually state on paper that if we want to understand things like shape and patterns and dynamics in biology, we really need to combine approaches from physics with biology, because these processes are fundamentally governed by the law of physics that apply to cells just like they apply to anything else in the world. And so the specific shape change I got interested in during my PhD is maybe a little bit niche. So this is what it looks like. So Michel, before I joined for a PhD, showed me a movie a little bit like this. And this is a cell, and this is a fragment of a cell. So it's not a whole cell, it's just a tiny piece of a cell, where you have done something to them so that their surface becomes more contractile, and I'll come back to that. But the interesting thing about it is that both the entire cell, but also the little isolated piece of the cell, have this kind of weird oscillating behavior, where they kind of change shape in a periodic fashion. And that's something that Michel had observed as a sort of random observation in 1989, and hadn't managed to convince anybody in his lab to follow up until 11 years later, when he trapped me in his office and showed me that movie. Actually, it was not this movie, it was a previous version of it, that doesn't matter. And I thought this was very interesting for a number of reasons. First of all, because you could see the same thing in a cell and in a small piece of a cell, that suggested that maybe there was something fundamental about biology happening here, because even a small fragment of a cell, without the nucleus, without those regulatory pathways, was able to display this behavior. And then from a physicist perspective, this was oscillating, and oscillations in physics are usually a signature of something interesting. There's a lot of oscillating systems in physics. They don't do this by random chance. There's usually an underlying network of, in this case, mechanical forces. And so that made us wonder whether there was something going on here which could tell us something broader about how cells control their shape in general, and not only in this kind of small feature of a cell that you place a little bit out of its comfort zone and make oscillate. And so what I spent the following roughly 3 years doing is trying to understand the mechanism of that. And when I say mechanism, for the more biology-oriented people among you, I tend to mean something broader than what's sometimes meant in molecular biology. So I don't necessarily mean a pathway, I mean how it works. And it might be a formula, it might be a physical model, or it might be a pathway, depending on the question we're looking at. And so in terms of how it works, I just show you the falsification of the hypothesis process in the thesis. So Michel actually thought since the 90s that this is maybe how this is working. So you have to see that back then, for people who know about actin, cytoskeleton, and cytoskeletal behaviors in cells, back then, the 90s was the golden age of the lamellipodium, which is a cellular protrusion which grows because an actin network pushes on the membrane, it's very flat, and it's very much driven by long filaments of cytoskeletal material inside the cell, which are these polymers that the cells form to support the shape, acting against the membrane to push it forward. So people were very focused on polymerizing networks being able to extend protrusions in cells. And so because of that, Michel thought that maybe these oscillations rely on such a network of parallel fibers of actin, for example. We knew it was actin-driven. And then something, probably a myosin-controlled ring, because it looks like a contractile ring, so it looked a bit like a dividing cell, is sliding on that. And so that's what he hypothesized, is maybe there's a sliding going on some sort of pre-existing network, which then turns over for some reason. And what we ended up demonstrating after a number of experiments trying to prove this and showing something completely different, is that actually what was going on in there is no sliding, but it's just contractions. And that's going to be a topic for the rest of my talk, because this contraction at the cell surface is still a large part of what we're investigating in the lab. And so essentially what's going on in these cells and the fragments is that most cells actually, most mammalian cells, or most actually animal cells, and even amoeba like Diptyostylium, have under the membrane a network of actin and myosin, which are basically a network of cytoskeletal filaments with molecular motors, which are called myosins, inside them, which exert contractile forces. A little bit like a muscle, except, and I'll come back to this later, except it's not as organized as in a muscle. So you have to imagine that there's a shell of contractile material underneath the membrane of pretty much any animal cell. And because it's contractile, it generates a pressure inside the cell. So the whole thing is under pressure. So it's as if there was a sort of contractile sock constantly trying to contract the cell. And it's not collapsing because there's osmotic regulation of volume. And then what happens is whenever you have such a contractile shell, if there's an instability somewhere, if it locally breaks, then the rest will squeeze and essentially make this kind of outburst of membrane without anything supporting it anymore, which will expand. And then the third part of the puzzle is that because everything in cell is dynamic, everything turns over. At some point, this burst membrane here, which is called the bleb, reassembles an actinizing network again, so cortex again, and then that goes back to the round shape. So this is what we proposed at the end of all this as a mechanism for why these cells and fragments oscillate. But more fundamentally, what this really drew our attention to is that maybe the fundamental biological feature that was at play here, which is actually quite important, it turns out, for the control of shape in many, many situations, is this idea that the inside of a mammalian cell is under pressure. And so that's something that in plant biology had been obvious for a long time, and also in bacteria studies, because they have turgor pressure, which is huge. And you know that if you remove the cell wall, a plant cell will literally explode. In mammalian cells, this was not necessarily considered to be very important because they don't have a cell wall, they don't have turgor pressure per se, and whatever pressure they have inside is not going to be very high, so it wasn't clear if it was, I mean, people didn't pay much attention to it. But what we realized, and as often in science, and those in other labs realize in different contexts about the same time, is that actually the inside of the mammalian cell is under pressure because of this contractile network at its surface. And again, to reiterate what I just said, that means that whenever there's a weakness and a local disruption in this contractile network at its surface, it's going to make a protrusion. And that type of pressure-driven protrusion is called a bleb. Which is not a great name, but it's been called that for 100 years, so there's not much we can do. So essentially, these oscillations and blebs in general, and that's a little bit of a crazy blebbing cell, are nothing more than a signature of the fact that the inside of the cell is under pressure. So another thing is that back then actually, blebbing in general, and I show you some examples of situations where blebbing is actually useful for a physiological process, but back then blebbing was still considered as something relatively bad, because this is what a cell would look like if you did a lot of bad things to it, like shone a lot of UV light or something. It's typically an apoptotic reaction. But as became apparent since, actually it's not necessarily a sign of cell damage, and there's a lot of cells which do that in all kind of physiological circumstances. But during the first maybe 5 years of having my lab, I actually have to start every single talk by explaining that what I'm going to be talking about is not apoptosis, and that these are actually healthy situations where cells do these kind of pressure-driven behaviors. So after that, I started my lab at the Max Planck Institute in Dresden, in Germany, which is this amazingly designed building, fantastic place to work, where I was actually co-affiliated with the Institute of Molecular Cell Biology in Warsaw, so it was kind of a double Polish-German appointment. And I spent 7 years there. My lab was up there, third floor. And this is the first lab. We looked young and dynamic. And the general thing I was interested in, building on these oscillation observations and all these things I had done at Curie, was to kind of go beyond that and try to understand, so that's actually fished this out from my job talk from back in 2005. What I became interested in, and in a way, as you'll see at the end of the talk, I'm still interested in, is what is the kind of shape space? What, given the physical properties of their surface and of their inside and of the environment they live in, what kind of shapes can cells display in general? And how do they navigate this shape state? How do they, in some cases, look like this and in some cases look like that? And then in a more narrow kind of depiction of this question, what we were planning to do is investigate how changing tension at the cell surface, so you could replace this by pressure inside the cell, can we actually transition from cells which would be round? I don't know if there's a way to increase the light on this polarity. I think there. Well, we'll see. Maybe there's not that much fluorescence. Can we transition from cells which are rather round and or whatever shape, epithelial shape, so some, oh thank you, which have some sort of stable shapes without many protrusions and deformations, to cells which maybe form blebs all over the place, to maybe cells which actually either oscillate or form stable rings like what you see in cell divisions. So are there mechanical parameters that we can change and tune this way and that way to make transitions in the kind of shapes the cells display? And one of the first systems we investigated this in, I'll just give you a snapshot of one of the first publications from the lab, was cell division. So we became interested in cell division because of the oscillations I had been studying, because the oscillations were essentially a ring, like in cell division, except it was moving. So we spent a lot of time looking at dividing cells, and that made us wonder, obviously this is an old question, how does that work mechanically? People have been wondering about this since it's been observed in 18 something. What the question, just looking at it from a very physics perspective, however, was how come it's stable? Because you do have a contractile cortex everywhere, and then it accumulates in the middle where the cleavage furrow will form and the cell will divide into two. However, there's still a cortex at the poles of the cell, even though it's weaker. So that means you have strong forces here and weaker forces here. But that doesn't look very stable, because either you maintain these two sides exactly balanced throughout the process of division, or there might be situations where one side becomes slightly more contractile than the other, but then this would tend to contract and possibly even collapse. And actually, from a pure kind of pressure perspective, and again, a cell is not a soap bubble, it has a spindle and all kinds of things inside, which might prevent that. But if these were soap bubbles, actually this would happen all the time. The small one would collapse into the big one. And that's something called the Laplace instability, which is an old instability in physics. So the question we kind of set to investigate is this shape stable, mechanically stable? And if it's not mechanically stable, what does the cell do to prevent this kind of instability collapses? And this was the work of, to highlight interdisciplinarity again, this was the work of my first two PhD students, Kuba Sentinski, who's a biologist, and Mate Biro, who's a bioinformatician. Kuba did all the experiments, Mate, or most of them, Mate did all the analysis. So one thing in physical biology, which is quite often the bottleneck, is analyze cell shapes precisely enough so we can actually say something. And that requires a lot of segmentation, tracking, all kind of things that are becoming increasingly available, but back then at least there was not much published about how to do this sort of thing. And then we've been very privileged to work with a fantastic theoretical physicist called Guillaume Salbreux, who was back then an independent postdoc in Dresden. He's now at the Francis Crick Institute, where he's had his own group for a while. And he's been my theory collaborator for the past almost 15 years, and he's a fantastic person to work with. And the conclusion of that study, which combined really precise measurements and experiments with physical modeling, was that actually, I'll just show you this one movie, this shape is not that stable. And that there's a lot of things one can do to destabilize it. And when you destabilize it, funnily enough, it starts oscillating, just like my weird fragments in my PhD. So this is one such condition where we depleted a protein which normally controls the recruitment of actin and myosin to the equator. So we basically make the poles stronger and more contractile. We can also trigger this mechanically. If you perturb one pole by, for example, shooting a hole in the cortex with the laser, which is maybe a bit brutal, but without destroying the cell, then you get the same thing. The cell starts oscillating. And since then, many other labs, and we have also followed up on a few of these studies, and we have essentially found collectively about 20 or 30 different proteins that when they become depleted, this is the kind of instabilities you get. So what that tells you is that this shape, even though normally this doesn't happen, is actually rather unstable. And I'm skipping the actual details of how we supported this with a model which was based on our experiments. But actually the conclusion was that mechanically speaking, from the cell surface perspective, it is just fundamentally unstable. And the cell has to stabilize the symmetric shape of the dividing cell to allow division to happen in a symmetric manner. And that led us to actually generate our first properly experimentally documented now state shape phase diagram or shape state, so to say, which looked a little bit like in my job talk, but with actual real parameters on the axis, and I'm skipping the numbers, but essentially the outcome of this, so it's a model, it's a physical model, so it doesn't mean to describe the cell in its entirety, but it does describe pretty well the phenomenon we were observing. Basically, what this model told us is that there is a whole region of this kind of physical phase space where symmetric division is stable. And this happens where tension at the pole is not too high, and where turnover speed of the cortex is, turnover speed is, if it's not too high, it's better. Or if it's turning over faster, so it's better. But there is a whole region, as tension at the pole increases, where you start having this oscillating instabilities. And that's something we expect, and I still hope to follow up on this experimentally someday. Something that the model predicted is that if you reach a certain parameter space, then maybe what could become stable is asymmetric divisions, where one daughter cell would be smaller than the other. And so that's, of course, a bit of an oversimplified view of how asymmetric division could be achieved. However, there's been a couple of papers since then in neuroblasts which suggest that this actually might be at play, at least in some developmental asymmetric division. So that was our first approach to phase diagrams of cell shapes. We also did other things, such as, for example, try to rehabilitate blebs. And we, for instance, investigated the roles of blebs in cell migration in a number of systems. This is a cancer cell, which is moving in an in vitro environment where we've confined it under agar, and you see it's bled all over the place. And this is a cell in vivo during zebrafish development. That was a great collaboration with Carl Philipp Heisenberg, who was also in Dresden back then, he's in Austria now, where together with our student Albert Munoz, we looked at how mesenodon progenitors in zebrafish, how they actually move in vivo. So this is inside a developing fish embryo where we labeled one cell. And what you can see is that the cell, as it's moving, this is the segmentation, is making all kinds of protrusions, including blebs. And so what Alba showed in her PhD, and then we have a couple of papers on that, is that blebbing is actually heavily involved in the migration of cells during embryonic development, at least in fish, but that's evident in other organisms. So that was the Dresden part, and then I moved to University College London in 2013, where I worked for about 6 years at the MRC Laboratory for Molecular Cell Biology, which is at on the main campus at UCL. And so I moved because for a number of reasons, but part of the reasons is that in the way the Max Planck Institutes work is that junior appointments are for 9 years max, and that's it. So there's no, you can't stay. I was not planning to stay anyway, but that's another story. But so I moved to London, and so one other common theme for students thinking about academic careers is I think another important thing is that in fact it's not bad to move every now and then as a lab, because you get new influences and that makes you do new things. So you're quite often scientifically you're very influenced by who's around you and what kind of, because of the people you talk to, but it's also because the kind of the kind of tools that are around you that might influence what you will do. And so, to a great extent, because we moved to the LMCB, which was establishing a very strong super-resolution microscopy facility just about at that time, I really became interested. So we still continued our work on like blebbing in all kind of things and cell shape in general. But one thing I became particularly interested in was going more molecular. So we had been working for a while on this scale, which was how the physical properties of the cell control what the cell look like, and why, we didn't never look at neurons, I'd like to, but why some cells look like this and some cells look like that. But then all this is controlled ultimately by molecules, and more specifically quite often by actin and myosin and this kind of cytoskeletal networks. So the problem we become quite interested in is maybe not quite from here, but how do you go from here to here? And the fundamental issue in addressing this problem is that ideally you would like to visualize these molecules and see what these different cytoskeletal networks actually look like at the nanoscale, and that's difficult. But that's where approaches such as super-resolution microscopy and electron microscopy come in quite handy. And so to make a schematic of the question behind this, so I've been talking quite a lot about contractility, so ignore the middle, just look at this one for that. Contractility at the cell surface. So what this is in blue, what the actin cortex look like if you take a snapshot with confocal microscopy. So there is a layer of actin at the cell surface. This is a dividing cell. And we know it has myosin motors, and we and others, and the field in general, tends to draw it like this. Like we have this random network of vaguely, not specifically oriented filaments, and then some motors which exert forces there, and that generate force. So, and that's not very easy to understand. Why is there contractile force in such a network in the first place? So the actomizing contractile network we understand very well is the muscle fibers. And their actin and myosin are arranged in a way that's almost perfect for contractility generation. Everything is aligned. The myosin thick filaments, so they are bigger than the ones you have in non-muscle cells, connect two separate actin filaments here in red. And when myosins walk, they walk towards the plus end of actins, which are here. So basically the whole thing is going to contract. So once people realized that this is what a muscle fiber looks like at the nanoscale, then it was quite clear why this was contractile. It's not like this for the cortex in non-muscle cells, because as far as we know, when I will show you some pictures of what it actually looks like, as far as we know, there's no order. It's really completely isotropic and random, and yet we know it's contractile, because we can measure this contractility. We can cut it with a laser, it's going to snap open. You can contract it, it's going to resist. You can kind of try to squish it under, for example, an AFM tip, and it's gonna resist this squishing in exactly the way you would expect for a contractile material. You can measure contractile tension in a number of ways. You can aspirate it in a pipette. And so we know it's under tension. We still don't really know why. And the reason we don't really know why, there's a lot of models. It's because we don't know how these things are arranged in space. So we draw these schematics, but we really don't know what it looks like. So that's what we've become increasingly interested in over the last 7 years now, is what do these networks actually look like? And just to highlight the problem, so this is again a confocal picture of a mitotic cell. The membrane is here in red, and actin is here in green on this overlay. I apologize for the color blind, I think I'll do better in later slides. And so what you can see is that there's a layer, but if you try to measure something even as simple as its thickness, you won't get anything because it's under the resolution of a confocal microscope. So actually now we know from a number of studies by us and others that this network, which is under the plasma membrane, is about 200 nanometers. So the resolution of a good confocal microscope is maybe 200 nanometers, the real resolution, the one you can actually get if you take a picture. This is what it looked like in normal electron microscopy. So this is the side of such a cell, and this kind of dark layer is the cortex. So again, okay, we can see it's there, but that's about it. Not telling us anything about orientation and arrangement of anything. So what can we do about this? Well, we can do scanning electron microscopy. So you can come to the surface of such a cell, remove the membrane, and then do some things to enhance contrast, and then do what's called scanning electron microscopy, which will tell you what it looks like from the surface. So that's better. Now we can see there's a network. That's the surface of a mitotic cell. These big cables are, so ignore them please, they are these kind of philopodia or microvilli, whatever you want to call them, which most cells have at their surface. But the key thing is this layer of this mesh of cytoskeletal elements here, which you see. So now we actually can see, and that's been known for a while, the scale here is 100 nanometers. So it's a very dense meshwork. The mesh size is maybe 20, 30 nanometers, which is really quite small, and I come back to that. It's quite dense, and as far as we can say, it's quite isotropic. Every now and then you see a clathrin coated pit, or all kind of blobby things which we don't know what they are. But these filaments are, as far as we know, active. So that gives us the surface. That's good. Can we go beyond that? Because if we really want to understand force generation, we need the 3D. So we need to know what it looks like across. And so I'll just give you a few snapshots of what we're trying to do about this. At this point, the short answer is at this point, nobody has been able to actually look into that properly. What we're trying to do is there's an electron microscopy technique called cryo-electron tomography, some of you may be familiar with, which essentially puts a sample in an electron microscope and turns it, and then collects many pictures and then reconstruct a three-dimensional tomogram from these many pictures. So that's great. It's done wonderfully at the NMB next door, for example. The problem is that to actually do this well, your sample cannot be much thicker than maybe half a micron, and that's pushing it, so 300 nanometers. A cell is 10 times that size. Is that right? 100 times that size? 100 times that size. So anyways, way too, you can't put a cell in there and see anything. The contrast would be, you wouldn't see anything because the cell would absorb too much electrons in the process, and you wouldn't see anything. So as a first step towards understanding what actin organization looks like at the cell surface in these round kind of cells, we decided to use these protrusions I showed you that form when pressure pushes them out, which are called blebs. Because turns out you can shear them off and isolate them, and they reassemble the cortex, and they are small. So it's not a cell, but it's a bit of cortex in a membrane. So it's as good as it gets for now. And then you have some ideas I can tell you about in a second about how to do it on an actual cell. But you can do that. So you can acquire a tomogram of an isolated bleb, and we see filaments in it, so that's good news. That's just to show you what kind of contrast you actually get, which is not, so here comes the problem of image analysis. How do you extract filaments from that? But one reassuring thing is that if we measure these filaments, they are about between 7.5 and 8 nanometers in cross section, which is exactly the diameter of actin filaments. So that's very reassuring. That will tell us that it's probably actin. Also, we can do a bunch of other controls to check that it is actin. And that's what a reconstruction of such a thing would look like. So there are now softwares that you can use to go from such a picture to a reconstruction of the membrane and the actin filaments inside with a relatively good degree of accuracy. But again, from a student perspective, when you see something like this, don't just take it for granted. It's only as good as your processing algorithm is. And so you may have missed things. And there's something you can, for example, if you try to measure lengths of filaments in there, it's usually not a good idea because the hardest thing in such segmentation is to figure where it ends. So the lengths might be quite wrong actually, but other things will be right, such as density. So we can do that, and so what we're trying to do now to achieve this in an entire cell is you can combine this kind of high electron tomography with something called focused ion beam milling, where you essentially take a big cell and then use a focused ion beam to destroy everything except a very thin slice in the middle of the cell. And then you have a sample that's small enough to use in cryo-electron microscopy, but that comes from the cell. And so that's what we hope to do sometime soon. So I'm not going to go to this, but that's just to highlight that using this sort of segmentation, you can now calculate all kinds of things, like density, cross-linking, contact points with the membrane, and so on. And now that gives us a better idea of what the cortex actually look like. But one last thing about these kind of nanoscale questions. One somewhat worrisome question that came to our mind when looking at these electron microscopy pictures and realizing how dense these contractile networks were, is that actually there was a problem when you tried to imagine how things that actually generate the contractile forces, like the myosin motors, how do they fit in there? Because this is what the myosin mini filament looks like to scale, drawn on such a network. So this idea that we've all been kind of accepting without thinking about it too much, that myosins just come to the cortex and diffuse through and get where they need to get, is quite a bit challenged by the realization that being so big compared to the mesh size is not very likely that they will diffuse very easily. That there's essentially a steric problem about getting anything through this mesh wave. So what we're trying to do now is actually visualize not only actin, but also other key components of contractility generation in the cortex, using less complex techniques than electron microscopy, or at least less complex to use. And for this, we've been using quite a lot something called super-resolution microscopy, and for example, structural elimination microscopy, which are relatively new microscopy techniques which have resolutions which can be as good as 20 nanometers, so 10 times better than the confocal, not for this one, for another type called storm, in good conditions. That means in real conditions, maybe it's going to be 50, but it's still, the problem is, again, coming back to this picture, is that 300 is about the resolution of the confocal. So if you want to see anything with the confocal, you won't see. But if you use super resolution, now you can see something. And so the very nice thing about myosin being so big is that actually you can visualize individual myosin mini filaments inside the cell. So for this, what we do is we label, for example, the heads in green like this, and then we look at the bottom of the cortex in structural elimination microscopy, and you start seeing, whenever you see a green, red, green, green, red, green kind of succession, that is a myosin mini filament. And you can do this live, so you can actually look, you can try to figure what the actual nanoscale organization of these molecules is. And again, to highlight that this is easier said than done, this is the kind of imaging pipeline you need to implement to actually extract anything from these images. But that can be done. That's why we also have people who know how to code and analyze images in the lab. But the whole question behind this is to actually, what we want to do is go towards a really understanding what the nanoscale organization of all these different components of the cortex is, and how they interact with each other in this very thin layer, which seems to be controlling what a cell essentially looks like. And the hope is, and then I just have 5 more slides, the hope is that we can combine this kind of super resolution imaging of different components of the cell with whatever electron microscopy can tell us about what these networks look like, and then use all that as actually measured ingredients and not just kind of hand-wavy ingredients into, that's a simulation now, into computational models of how all these things interacting together actually generate contraction at the cell surface. And how do you do such a thing? How do you go from a sphere towards a cleavage furrow? And this really happens by, in the first place, by building higher contractile tension here at the equator, and then the cleavage will just happen naturally once you have built that. So that's towards combining physical and biological mechanism, if you want, because we really want to know what the molecules are doing. And so in the last few slides, what I'd like to do is just highlight towards what we are going now. So as you heard in the introduction, I moved to Cambridge in 18 actually. Anyway, mostly because of this little person here. I mean, partly because I got a fantastic job, but also because my partner has a lab in Cambridge, so we were trying to reunite. So these things happened in a very convenient sequence of time. And I moved into the Department of Physiology, Development and Neuroscience. And again, because being in a new environment always inspires you, it didn't actually trigger, we were thinking about this before already, but it kind of solidified another direction that I've been wanting to expand the lab towards, which was what's the coupling between actually cell fate and cell shape? So I showed you this first slide highlighting that all these different cells in the adult organisms do different things and have different shapes. And so it appears, based on a number of studies, that there's actually feedbacks between shape mechanics and what the cells do in terms of fate and state and differentiation. And so being in a department where a lot of people study development, and being in actually university, which is very strong in studies of development, stem cells and evolution, I come to that in the last slide, that really made us go more towards how can we go about understanding how cell fate and cell shape are coupled. And so one system we've been looking at this in is exit from naive pluripotency, so very early differentiation of embryonic stem cells. And that's because this is a stage where cells change shape quite dramatically. So on the left is a picture of a naive embryonic stem cell from mouse. And this is, so you can keep the cell naive and self-renewing in a dish for, well, ever is a long-term time, but as far as we can, it can be kept naive for a long while. It can be also put back into a mouse embryo and it will contribute to all the tissue, so it's truly naive. But you can also induce exit from naive pluripotency and then further differentiation in a dish. And if you do that, in about 24 to 30 hours, the cell looks like this. So actually I'm lying, this is not one cell, it's probably 2 cells stuck together, and here you also probably have 2 cells, but they adhere very, very tightly. And these are electron microscopy pictures that we kept the membrane, so they look pretty. And the shape change is really quite characteristic. So these cells are somewhat isolated, they are often in colonies, but they usually display this shape change. And so we had a study which is just on my archive for the last few months, giving some ideas about how the mechanical changes underlying this transition actually affect the signaling pathways that control differentiation. But what I'd like to highlight now, coming back to my general theme of my interest in understanding what the shape space is you've been using this transition as a kind of prototypic transition from round to spread, which happens a lot in all kind of situations in biology, to try to figure a pipeline to do some sort of morphometric analysis of cell shape changes. And what I mean by that is now we can essentially take a lot of naive cells and take a lot of exiting cells and then segment them, and then you can extract a number of parameters. You can do it by hand, like this. You can measure the area, you can measure the solidity, so how spiky and not spiky they are if you want. But what we wanted to do really is something more global than this. So we didn't want to look just like at 5 or 10 parameters that would describe these shapes, but just as many parameters as you can, and then can you put them in some sort of multidimensional reduction space? Just like for those who are familiar with this type of approaches, people do this a lot for gene regulatory networks. So you can do what you would do typically in stem cell science. You would do a single cell sequencing on these cells, and you would have all the genes expressed in this type of cells and all the genes expressed in this type of cells, and you compare them by doing principal component analysis, for example, which is a way to reduce the multidimensionality of this data into a subset of parameters to be able to plot in a graph like the naive cells live here and the differentiated cells live here. So what we've been attempting to do is to add parameters of shape and mechanics to this sort of approaches. And so essentially you can take, I think we take 16 at this time, but you can take whatever number of parameters you want along the lines of area, diameter, log axis, spikiness. You can make it more fancy and put some Fourier descriptors in the picture, and then condense them by doing what's called principal component analysis, which essentially consists in projecting all these parameters in 2 dimensions. And so now that allows us to see how shape changes as described by these 2 parameters, which are not just area. It's a combination of all these different 16 parameters we've been looking at with certain weighting. And now all the naive cells are in pink, they are here. And then as they exit nitrogen potency and become increasingly spread, they transition to a dispatch of this shape space. So this is early days. We're trying to do this in a number of systems, and we try to see, so the aim of this is now to, for example, put expression levels of key markers of differentiation on these little dots and see if these are more differentiated than these, for example. Just to understand to what extent this transition in shapes goes together with the transition in what these cells are actually doing in terms of gene expression. And the idea is basically use this to add a third axis to this thing. So what we've been studying for a while in the lab is how mechanics affect shape and organization in individual cells, also a little bit in tissues. I didn't touch upon that. But now, in fact, cell fate decisions are primarily controlled by signaling and gene regulatory networks. But these also control mechanics. And actually, there's feedbacks from mechanics to these. And there's also feedbacks from, there's fate is associated with changes in mechanics, but there's also feedbacks in all directions. So that's the grand scheme of what I'd like to understand in the next 30 years. But to finish with some pretty movies, one last kind of weird direction we became interested in is I've been always kind of wondering about, okay, how about evolution? Let's get a bit crazy. So I heard a talk by Mark Kirschner, who's an amazing scientist from Harvard, very, very prominent in my field, who mentioning, he was giving a talk about his life at the conference, and he said something along the lines of, when you become interested in evolution as a cell biologist, that means you're really getting old. So I don't know what that means about me, but I hope he's wrong. But we kind of, so I've been interested in trying to understand how cell shape evolves and how physics maybe affects that for a long while, having done nothing about it, because something which is, I don't even know how to study this properly, but something which is quite obvious is that physical constraints have to affect evolution in some way, at least evolution of shape. Because shape is about mechanics, so the way mechanical forces are controlled have to affect the way cells can evolve in terms of shape. And so kind of to have a go at this, and this is really the beginning of a project which started in September, and it's a PhD project of Margherita Battistara in the lab, who is co-supervised by Matt Benton, who is in the zoology department, and he's interested in evolution. And what we're looking at is essentially, we want to use this pipeline, this morphometric analysis of cell shape transitions, to compare how cell shapes change during gastrulation, which is a process where cells transition from being epithelial or somewhat rounded to being mostly somewhat spread. So again, this round to spread transition in 3 different insects for reasons of feasibility. That's just some very large scale movies. These are the entire embryos of, this is a Drosophila embryo undergoing gastrulation. Gastrulation happens here, all these cells go inside, and actually once they form this kind of impression here, the cells will start spreading inside the embryo. This is, I will start with this one, this is Tribolium, it's a beetle. Again, it's undergoing gastrulation, there's a whole tissue going inside another tissue, and the cells on the inner side will spread. And this is a moth, which is a new experimental system, and this is one of the first movies of the dynamics of cells in such a moth that Matt has been looking at how to study that in Woods Hole recently, and so that looks very exciting because contrary to the other two, it's very easy to work with. And it doesn't eat cloves. I think this one just eats cabbage or something like that, so that's good. And so what Margarita has been establishing, and that seems to be working pretty well, is essentially imaging technique to really look at how cells spread inside this embryo. And for example, that's a movie of the segmentation of the different cells. It's a 3D kind of movie going back and forth. And so what we're planning to do is essentially properly analyze using our pipeline of like morphometric analysis how cells change dynamically in these 3 different insects which are separated in evolution. And then do comparative analysis to understand to what extent it's the same process, it leads to the same thing. And it's similar shape changes. But to what extent are these shape changes really similar? Are there differences? And then, if we have time, or maybe that's another PhD, how's that controlled? Because of course, if you talk about evolution, some pathways must have changed if they are different. And so what we'd like to understand is how, because we know a lot of things about how shapes, what controls specific shapes in terms of cytoskeletal networks. So how these different species use different aspects of actin mechanics to control their specific shape changes. And again, the big question behind this is, what's the kind of shape cells can have, and why do they sometimes have this type of shape and sometimes another? And I'll just finish with a few thoughts for the students about interdisciplinarity. So first of all, it's really important because we tend to be taught in biology or chemistry or physics or whatever, you study what you study, but cells and organisms and living matter in general doesn't care very much. They use all of these things. Obviously they use chemistry, biology, if one is extreme, is in a way a combination of physics, chemistry, and all these other things. With its own laws, maybe, that's an open question. But so I think it's becoming increasingly important in research, and it's quite clear if you look around you at what people are doing in different labs to actually combine approaches from all kind of disciplines. So either one can attempt to be trained in really at the interface, that's one way to do it, but actually doing it in collaboration is totally fine, and it's great, and it's lots of fun. But that still involves really going out of your comfort zone, so you have to be not afraid of doing something that you maybe don't fully understand at first, but ask questions. And if your theoretical physicist collaborator can't explain why the equation looks like what it looks like, you don't need to care about how he solved it, doesn't matter. But the point is you have to know why the equation looks like what it looks like, and if they can't explain it, then that's probably not the right collaborator. But usually if you push them enough, they will be able to explain it. And then it's good fun. And then you will explain to them that biology is not as simple as they think. And that's just a couple of papers I wrote with my general, very limited thoughts on the matter. But if you're interested in this, go have a look. And finally, just an announcement, you may be aware, we recently launched, literally last week, I think, or 2 weeks ago, Cambridge Center for Physical Biology, which is a virtual network of events, seminars, all kinds of things, trying to connect better physics and biology across Cambridge. There's actually a lot of labs doing interdisciplinary research, but we feel we're not connected enough as a whole, and so we're trying to develop that. And I just would like to thank my labs over the years, and I won't actually single out people because there's been many things I talked about in this talk, but that's just some pictures. That's from Dresden Times, that's from London Times, Cambridge Times to come, and I'm also grateful to all these people for giving us money, and thank you very much for your attention.
Okay, thank you, Professor Paluch, for the insights, especially about interdisciplinary science. We have a couple of minutes to take some questions, so yes, please.
Hi. Thank you for your talk. It was very lovely.
Thank you.
I have a couple of questions. So you can let me know when somebody else has a question or whatever. But just starting from the back, so towards the end. So when you were using PCA, what was the principal variable? What did you make to make it principal? What was the thing that I let it be identified as this was the principal component?
So you mean how we chose 2 and not 3 and
Yeah.
not 5? So I think in the end we used the variance across the different principal components, and it happened so in this case that most of the variability in shapes was described in the first 2. So you choose them by basically how variable they are between the different cells you're looking at.
But were you using different types of cells?
Yeah, no, so actually the way it works is you pull all the cells together, and you look at the variability. So you basically project them in the mathematical. So these are mathematical variables.
Yeah, like in MATLAB. Yeah.
But so it combines all the different parameters in one number.
Mhm.
It's a bit different way here as an explanation, but basically it's a way. So the first principal component, if you want, it would be a combination of the area and the height and the spikiness, but with different weightings. So let's say, I don't actually know in this specific case, but it might be mostly area, let's say, but it could be also have a component of how elongated it is.
I see what you're saying. So then in the example you just said, then the most principal would be the weighted, since that was the one that was different.
Yeah, exactly.
Okay.
So basically there's different contributions of the different parameters to that.
Okay, and then I have questions about blebs. How do you get the blebs? Were those just general models that you're using to illustrate your point?
Oh, you mean how do you get them to
No,
isolate them?
like how did you specifically get the blebs? Because I know like I know that some groups have to make protocols to get blebs from certain cells. Yeah.
Yeah.
And like I know like there's only a bacterial cell line. There are some bacterial cell lines that do blebbing on their own, I think. But I was just wondering
they trigger blebbing.
Yeah. So I was just wondering how in particular did you get those, or was that just a model just to illustrate your point?
Yeah, it's in a way a model. So we make them in a somewhat artificial way. So we destabilize the actin cortex, and then we literally shake, and they fall off. With a drug.
Oh, okay. You with a drug.
Yeah. So
like
there's two
chemical.
things you can do. You can use a cell which is naturally blebbing a lot.
Yeah.
These are called M2 cells. They don't have much filament. It's a cancer cell line,
Mm-hmm.
which is very blebby naturally.
Mm-hmm.
Or you can take any cell line and destabilize actin with a drug that depolymerizes actin, and that's going to make it bleb all over the place.
Okay.
And then you shake them off.
Okay, and then hello. Oh, you have one? Oh, okay. Yeah, and so then when you talk about how they form, like okay, so I think you just answered that, how they form. So when you were talking about how blebbings indicate intracellular pressure, why does that indicate intracellular pressure?
Because it wouldn't form if there was no pressure. So it's really squeezing.
Okay.
So essentially the cell body squeezes, and if there's no weakness, then it just builds pressure.
Mm-hmm.
If there's a weakness, it's like sometimes I have this toy the kids have, like if you
Yeah, and then the little thing pops. Yeah.
That's exactly what it is.
Oh, jolly good.
Yeah.
Yeah, but that's why I'm asking, because like in those toys, like all they do is they don't pop off and become a little small thing. We call them blebs. They just go back. So I was wondering, like for that instance that we have pressure, but then it just pops up like that and it goes back. Or for the instances of the cells, it pops up, but then it becomes something else. So I was just wondering.
It would go back actually, but because in cells everything turns over. So what happens is that this actomizing networks I was talking about, they come back into the bleb and then they retract it.
Because it's because of actomyosin.
Yeah, so everything is dynamic.
Jolly good. So if that toy had actomyosin, it probably would be jolly good.
Exactly.
Maybe we can take some questions from
Oh, okay.
audience.
Jolly good. I have more, so
yeah,
you can come back to me.
yeah,
Jolly
yeah.
good. Thank you greatly.
Anyone else have questions? Yes, please.
I have one with the evolutionary story at the end.
Mm-hmm.
I was just wondering, wouldn't you expect rather maybe developmental pathways or some signaling just being differently used rather than the evolution occurring at the level of, let's say, the protein or fibers? That would be the same mechanism, just...
Yeah, I guess what I was trying to I think that the question we're trying to approach is really, there's something fundamentally important about cells spreading as they undergo gastrulation. So that happens in most organisms.
Mm-hmm.
So cells are initially either epithelial or somewhat epithelial, not necessarily very, very polarized, but mostly rather cuboid or round. And then as they become mesoderm, they spread and become migratory, and that's the general feature. And so in fact, the interesting thing about that is that this sort of transition also happens in cancer cells, but as you're right, controlled by somewhat different pathways, but at the level of actin itself, it's probably not so different, but that's the question in fact. That's what we want to understand, is whether actually there's different ways for the cell to spread, or whether these are all the same way to spread but controlled by different ways, and if it's controlled in different ways, then what's the constraints? How come that it still always spread? You see what I mean?
Yeah. Thank you.
And it's a very open-ended question.
Mm-hmm.
Any other questions, please?
So thanks, Ewa, lovely talk.
Thank you.
So I just had one question. So when you have these cells, you are saying that the contractility is sort of the same, and that's why the shape is being maintained. In cells which undergo endocytosis or exocytosis, so you have the local dissolving of the cortex,
Probably.
and that's how the membranes fuse. So does that not create a whole imbalance, and won't it?
Yeah, that's actually an excellent question, and that's very, very poorly. I don't know if I can pull out that slide again, but that's very poorly understood. So a big question related to the density of the cortex is if you try to make an endocytotic vesicle go through this, how does that, or a virus or a bacterium or whatever. This is a class 3 coated pit, I think. So it's quite big. So yes, you're right, it has to be solved locally, but nobody has actually really seen that. So partly because it's too small, partly because we probably didn't look properly, but that's a big question in the field right now. How does that actually happen? And how do you drive the membrane through the network? So it's poorly understood.
Do you have a question?
Yeah.
So I have a question. So what's the impact of environment and other cells around on the cell shape? Do you think this is mostly dictated by the internal structures itself, like actin, as you mentioned, or the environment? Because when you culture cells, especially taken from cancers, they all look pretty similar. So what's your...
Yeah, no, you're absolutely right. So I was giving a very oversimplified picture because it's true that in many of the studies we focused on individual cells, but cells in tissue and cells with neighbors and actually cells strongly adhering to a substrate, and that's where this transition to looking towards spreading actually interactions with the substrate become absolutely essential. So yes, so the small part of the mechanical picture I spend a lot of time focusing on is contractility at the cell surface, but then if you take a contractile sphere and make it stick, then you change the force balance, and that's obviously very important. And then in a tissue, a cell is being pulled and pushed and pulls to its neighbor, attached to its neighbors, and so on. So actually, these things are really, really essential. And but the concept of pressure remains important. And for example, there's a study on angiogenesis, so how you form blood vessels, which indicated that in at least a specific case, it seems to be driven by some sort of reverse blebbing. So the pressure from the outside pushes the membrane inwards, and that helps. So that's from Holger Gerard's lab. Helps forming growing vessels. So this concept of pressure and how imbalance of pressure can deform cells and maybe even entire tissues is important. But how adhesions play into the picture, and also a lot of other intracellular components I didn't talk about, like intermediate filaments and microtubules and so on, all these things matter. So it's much more complicated. Yes. Thank you.
Maybe we'll take one last question and then we'll hang around for more people to ask. Yes, please.
So in this shape phase diagram, there was this area of the asymmetric division. Could you talk a little more about this? Like, what would that actually mean? Does it occur in nature, or?
I think it occurs. I mean, asymmetric divisions occur. Many of them are because the spindle moved. So most of the time, asymmetric divisions occur, especially in development, because quite often, I mean, the obvious one is meiosis. So when meiosis happens in the fertilized egg, you want to keep everything in the egg and make a small thing leave with half of the chromosome or genetic material. But in many other developmental processes, there are asymmetric divisions. Very often they are result from the spindle being displaced. But there's at least one system, which are neuroblasts, so precursors of neurons, among other things, which are quite asymmetric in many species. And there's quite some indications. These things are harder to study properly in vivo because measuring forces in vivo is more difficult. But there's quite some indications that at least in several species that there's an asymmetry essentially because one, as your cell divides, one pole contracts more than the other. So that's what this, and why stabilized, that's something I don't understand physically. There must be something happening. But essentially it doesn't oscillate, it just squeezes slightly and you have one small cell and one big cell. So it happens, but how exactly is not very well understood.
Thank you.
Okay, thank you, Professor Paluch. Thank you.